So in prokaryotes, we talk about two main polymerases, DNA polymerase I, DNA polymerase III. So DNA polymerase III does most of the replication of the leading and lagging strands. DNA polymerase I essentially removes the RNA primers and fills in the gaps left when the primers are removed. Now, in eukaryotes, the polymerases involved in replication are polymerase alpha, polymerase delta, and polymerase epsilon. In the current model is that polymerase alpha, this makes the RNA DNA primer. Oh, there's another difference, right? The primer that's made in prokaryotes, well, okay, this is made by an enzyme called RNA primase. Now, polymerase alpha is the one that takes the function of RNA primase in prokaryotes. It makes an RNA primer initially, but then it switches to using deoxyribonucleotides. So it makes a hybrid RNA DNA primer. So really cool, right? It doesn't just use ribonucleotides. It uses ribonucleotides first, and then it switches to deoxyribonucleotides. Okay, so pol alpha is making the primers needed for DNA replication. And it needs to do that because delta and epsilon cannot synthesize DNA on a naked template. It needs a primer in place in a 3' hydroxyl group in order to synthesize DNA. Now, the traditional model is polymerase delta is in charge of lagging strand synthesis. And epsilon is involved in leading strand synthesis. Now there is a new model, and I'll just touch on that. And this new model suggests that polymerase delta is in charge of both lagging strand and early stages of leading strand synthesis before polymerase delta hands it off, leading strand synthesis, to polymerase epsilon. Now that's just, something very new. So what we're going to do is just focus on these as the major functions of the eukaryotic replicative polymerases. Now, another thing I wanted to touch on with respect to DNA replication in eukaryotes was the replication factor C, RFC, this complex right here, and PCNA, which is another complex. Now, these are thought to increase the processivity of the DNA polymerases, in particular polymerase epsilon. Okay, what does this mean? Processivity with respect to a polymerase is the, let's say, the number of nucleotides in a DNA molecule that the polymerase will, polymerize at one time before it dissociates from the template. So the higher processivity, the more DNA, the more nucleotides that are joined together by the polymerase before it falls off the template, whether it be accidentally or because the polymerase is finished. So if this is a replication fork, and it's moving in this direction, and let's say we have, this is a template, let's say we have polymerase alpha, epsilon right here. So traditionally, PCNA is shown as like being behind the polymerase and sort of holding it on the template, keeping it from falling off. So you can see how having this PCNA complex here associated with the polymerase would help keep it from falling off the template, and therefore increase the processivity, increase the number of bases joined together by polymerase epsilon before it falls off the template. It needs to be reloaded on. And replication factor C, now this would be another complex of proteins, and it's thought that replication factor C loads PCNA onto the template with the polymerase. So that's something new that we didn't really touch on with prokaryotic DNA replication. I'm not actually sure if there are equivalent proteins in prokaryotes. There might be. Okay, so what we're going to spend most of the time on in this video is the differences in Okazaki fragment maturation in prokaryotes and eukaryotes. So now, what is an Okazaki fragment? And I'm sure you watched the previous video, and I'll just review that here. So Okazaki fragments are synthesized during lagging strand synthesis. So they're short fragments, and they result because the lagging strand needs to be synthesized in fragments, because the direction of replication is opposite that of the movement of the replication fork. So I'm using these different colors here to indicate. So red is going to indicate let's say we're going to take prokaryotic replication, prokaryotic Okazaki fragment maturation first. Here is the RNA primer, the beginning of one Okazaki fragment. Right here, I'm going to put OK right there, and here is another Okazaki fragment right here. Here's another RNA primer right here. So you can see the RNA primers are at the 5' ends of the Okazaki fragments. Now in prokaryotes, what happens is DNA polymerase 3 synthesizes the Okazaki fragments, and DNA polymerase 1, it uses its 5' to 3' exonuclease activity. Now DNA polymerase 3 lacks this activity. DNA polymerase 1 has it, so it uses its 5' to 3' exonuclease activity to remove the primer starting from its 5' end and moving in the 3' direction. So it also uses its 5' to 3' polymerase activity to fill in the gap that's left after removing of the ribonucleotides associated with that primer. So it's going to use the 3' OH at the end of this Okazaki fragment. It's going to fill in the gap as it's chopping out these ribonucleotides. And eventually we're going to be left with something like this. And it's going to fall off the template. I'm not sure if anyone knows the mechanism by which it falls off the template or stops synthesizing, polymerizing DNA, but it's supposed to keep going until it removes all the ribonucleotides. And what we're going to be left with is a small nick. I'm going to put a phosphate group here, and an OH group here. Okay, so what is a nick? Now, I'm putting a nick is essentially just a break in the phosphodiester backbone of a double-stranded DNA molecule. So all the nucleotides are there. If any nucleotides were missing, that would be a gap. So all the nucleotides are there. There's just, we're missing a phosphodiester linkage between these two adjacent nucleotides right here in this strand. So what did DNA polymerase 1 do? It removed that RNA primer, it filled in the gap, and when it fell off, it left a nick. So this would be sealed by DNA ligase, and that's like that's like step 6 of DNA replication. It's for DNA ligase to come through and seal all those nicks that remain from Okazaki fragment maturation. Okay, now that's how Okazaki fragment maturation works, is thought to work in prokaryotes. Now, what about eukaryotes? So it's a little different in eukaryotes, and this model is relatively easy to understand. That's why I think it's a good one to focus on when we're talking about differences between prokaryotic and eukaryotic DNA replication. So Okazaki fragment maturation in eukaryotes, in eukes. So let's diagram this molecule again, or this DNA strand, this DNA template. Let me put in the Okazaki fragments. So now in eukaryotes, I said DNA polymerase alpha makes the primers, and it makes a ribonucleotide DNA nucleotide hybrid primer. The ribonucleotides are put down first, and then DNA nucleotides. So we only need to remove the ribonucleotides. So those DNA nucleotides, they can stay. So the red just indicates where the ribonucleotides are from that primer. So DNA polymerase alpha puts down the primer, and DNA polymerase delta extends the primers. Now, okay, so how does Okazaki fragment maturation in eukaryotes work? So DNA polymerase delta is the one that does most of the lagging strand synthesis. And let's say we have DNA polymerase delta right here. What it's going to do, it's not going to stop when it hits this primer. What the model is that it wedges itself between the primer and the template and dislodges a little flap. So we end up with something like this. So, and here's the flap right here. And the flap might be 2 to 3 nucleotides long. These are ribonucleotides. Okay, so once this flap is made, it gets stuck. It can't go forward anymore. It can't put down any more DNA, deoxyribonucleotides. So what happens is an enzyme called FEN1, also known as the flap endonuclease, comes in and it chops off the flap. So it removes the flap. And once that flap is gone, well, DNA polymerase delta can push itself in, wedge itself in again, and make another flap. So the reason it has to do this is it lacks a 5' to 3' exonuclease activity. You know, that activity that DNA polymerase 1 has in prokaryotes, well, DNA polymerase delta doesn't have it. So it's thought to help, it's thought to participate in Okazaki fragment maturation by wedging itself in here, creating the flap. FEN1 removes the flap, it wedges itself in again, and FEN1 removes that flap, and it keeps doing this. This process keeps going back and forth between FEN1 and DNA polymerase delta until all the ribonucleotides are gone. And then you can probably predict what's going to happen next, right? Just like with prokaryotes, we're going to be left with a nick. The with a phosphate group on the 5' end down here, and a hydroxyl group on the 3' end of this fragment right here. And that's going to need to be sealed by DNA ligase. Okay, so those are the major differences between Okazaki fragment maturation in prokaryotes and eukaryotes. And I think that's it for this video.